Okay, welcome. The sexual reproduction in flowering plants, second year, second chapter. See, reproduction is a biological process in which an organism gives rise to young one of its own type, and it enables the continuity of species generation after generation. It is a must. It is need of not only to the living organisms but to the nature, both in plants and animals. The young ones are formed from their parental generation, and as per the lifespan they will leading to or proceeding to the life and coming to the end. So the next generation is needed for the existence of that particular species, and so that is why we can say the reproduction is actually a unavoidable process in the life of living organisms. See here, the sexual reproduction in flowering plants. So plants have vegetative, asexual and sexual means of reproductions. Among them we are going through the sexual reproduction. So the asexual, vegetative, everything will be seen in the first chapter, that you can see later. And so here we are going through the sexual reproduction in flowering plants. See, sexual reproduction in flowering plants or angiosperms. So what is reproduction? Reproduction is a biological process in which an organism gives rise to young ones of its own, and it enables the continuity of species generation after generation. It is a general concept. Reproduction is a biological process in which an organism gives rise to young ones of its own, and it enables the continuity of species generation after generation. It is a must for the nature. The various kinds of living organisms aggregate to form a biosphere having living and non-living evolutions. Among them, the living evolutions needs their existence and their generations forever and ever. See, according to the plants, vegetative, asexual and sexual reproduction presents. See, what is reproduction? Reproduction is a biological process in which an organism gives rise to young ones of its own kind, and it enables the continuity of generation after generation. See, so according to the sexual reproduction, sex organs are needed. So there, for the sexual reproduction, two parental generation is also needed. So male and female parents, or male or female sex, whether it is on the same flower, same plant or in separate plants, that monoecious, dioecious, bisexual, unisexual structure that already learned in morphology, and so you can go through the first chapter also. Wait. See, the sexual reproductive organs in plants are, in plants means flowering plants, are flowers. Flowers are the sexual reproductive organs in flowering plants. The flower contains both male and female reproductive organ, female reproductive parts. So since flower is the carrier of male and female reproductive parts, they can easily participate in sexual reproduction, but an indirect way when compared with that of animals. That means pollination process is also there in the flowering plants or in plants generally. So the flower contains both male and female reproductive parts, and hence the sexual reproductive organs in plants, flowering plants, are the flowers. The male reproductive part is androecic. Male reproductive part of a flower, androecic, formed of simple units called stamens. They are the male sex organ. And then the female reproductive parts of a flower, reproductive part of a flower is gynoecium. Formed of simple units called carpel. The female sex organ. Carpels, the female sex organs. The stamen contains anther in which pollen grains are formed. Pollen grains are formed, in which pollen grains are formed, from which the male gametes, sperms are produced. See, stamen contains anther in which pollen grains are formed, from which the male gametes, sperms are produced. Like that, the carpel contains ovary in which the ovules are produced. Ovules are produced and the female gametes egg is seen within the embryo sac. And it is interesting to say that male contains male gamete, female contains female gamete, but their fusion results the formation of a zygote. So here an intermediate stage, the pollination followed by double fertilization leads to the formation of fruits and seeds that carries the zygote or embryo, that carries the zygote. The sexual reproductive organs in flowering plants are the flowers. The flower contains both male and female reproductive parts. They may be on the same flower if flower is bisexual. If in separate flower, they are unisexual flowers. The male reproductive part of a flower is androecium, formed of simple units called stamens. They are considered as the male sex organs. The female reproductive part of a flower is gynoecium, or we can say the pistil, okay, formed of simple units called carpels. Carpels are considered as the female sex organs. The stamen contains anther in which pollen grains are formed or developed, from which the male gametes sperms are produced. The carpel contains ovary in which the ovules are produced, and the female gamete egg is seen within the embryo sac. That comes later. The pollination followed by double fertilization leading to the formation of fruits and seeds that carries the zygote or embryo. Seed germinates to form an young plant called seedling. So seedling is the beginning of, or seeds are the end product of sexual reproduction, and from the seed there begins the next generation of a plant. See, so we can go through the reproductive organs like flower, and the flower contains two kinds of organs, helping organs and essential or sexual organs. Suppose the reproductive organs, flower. Longitudinal section of a flower carrying all the organs, and the flower is complete as it contains the four typical bulbs like calyx, corolla, androecium and gynoecium. And this is pedicel. This is thalamus. The outermost well, calyx, formed of sepal, corolla, formed of petal. These are helping words. These are helping words. And this is the male part, androecium, formed of simple units called stamen. And this is gynoecium, formed of carpel. These are the essential words performing the function of sexual reproduction. Thus, helping words enclosing the essential words or words performing the participation of sexual reproduction. Okay? And stigma style ovary that you can see later. While we take the LS or longitudinal section, the ovary carries the ovule and ovary becomes these are ovules. So stigma, style, ovary enclosing numerous ovules or a single ovum that you can see later, and the whole part is the LS of a flower, ground plan. LS of a flower, typical flower. LS means longitudinal section. And so, in a nutshell, what I am already given to you through a flow chart, how they are connected each other, and what are the other names in connection with the parts of a typical flower for the convenience of study. See, this is LS of a typical flower showing the four words, among which the essential words like gynoecium, androecium. Gynoecium is the female reproductive part. This is female reproductive part. Symbol for female, and this is male. Then the essential words. Androecium, gynoecium. This is female, this is male. Androecium is formed of simple units called stamens. Stamen is also known as microsporophyll. Stamen is also known as microsporophyll. Gynoecium formed of carpels. Carpel, also known as megasporophyll. Stamen contains anther. Anther is provided with a megasporangium. Anther contains pollen grains. Also known as microspores. On their germination, they become male gametophytes. Male gametophyte produces male gamete. Male gamete is also known as sperm. This is a stamen. In details you can see under filament. It produces pollen grains. So here carpel contains, this is a carpel. Stigma, style, ovary. Ovule, carpel, stamen. Carpel contains ovary, contains ovule. Megasporophyll contains ovule or megasporangium. The megasporangium contains megaspore. Megaspore develops to form embryo sac. Embryo sac is also known as female gametophytes, producing egg or female gametes. Female gametes or egg. Then here we know pollination. Followed by double fertilization. Giving zygote develops to embryo. Embryo develops in the seed. Seed on germination seedling is formed. This is actually the flower and connected sex organs and their details and how they are connected together. See, flower contains male part androecium, female part gynoecium. Androecium is formed of simple units called stamen, also known as microsporophylls. Microsporophyll stamen contains anther, which carries the microsporangium. Microsporangium. Microsporangium carries the pollen grains, which are the microspores. Microsporangium contains microspores. Sporangium contains spores. Microsporangium contains microspores. They are also known as pollen grains. On germination after pollination, pollen grain becomes male gametophyte. Germinating pollen grain is called male gametophyte. Gametophyte produces gametes. So, gametophyte producing gametes. Male gametophyte producing male gametes. So, male gametophyte produces male gametes. Male gametes are also called sperm. Like that, the gynoecium, female reproductive part, producing simple units called carpel or female sex organ. It is also known as megasporophyll. Megasporophyll contains numerous ovules, which are called megasporangia. The megaspore producing structure, megasporangium. Megasporangium producing megaspores. Megaspores develop to form embryo sac. It is also known as female gametophyte as it produces female gamete or egg. So the female gamete is produced in the gynoecium, male gamete produced from the androecium. So here the two processes, that is pollination followed by fertilization, producing zygotes, which develops to form embryo. Embryo is seen in the seed. On germination, seedling is formed, and seedling is, this is the seedling. Radical, plumule. Seed, radical, plumule. Radical develops to form root system, morphology. Then plumule develops to form shoot system. Thus a plant contains part in the ground and part above the ground. In the ground is the root system. Above the ground is shoot system. Shoot system carries more or less all of the parts of a typical plant. Leaves, branches, buds, flower bud, flower, apical bud, axillary bud, so many parts which are all developed, fruits, seeds, which are all developed generally in the shoot system. A root system fixing the plants, absorbing water and minerals, and support to the growing plants. And so, listen, flower, androecium and gynoecium, male and female. Androecium is formed of stamen. Stamen contains anther. Anther contains producing pollen grains. Pollen grain act as male gametophyte. Then male gamete, the sperm thus produced. So here gynoecium, the female reproductive part, carpel, the simple units. Then it contains ovule. Within the ovary, ovule producing structure is called megaspore. Functional megaspore becomes active later, and the megaspore contains the embryo sac, and embryo sac contains the female gamete. Embryo sac contains female gamete or egg. See, stamen is also known as microsporophyll, producing microsporangium, carries microspores, develops male gamete or sperm. Carpels, the megasporophyll, carrying ovules, producing megaspore, then female gametophyte, the embryo sac, and female gamete. So, megasporophyll is carpel, microsporophyll is stamen, megasporangium is ovule, microsporangium is anther. Microspores are what? The pollen grains. Here, the process of production, formation and development of microspores from microsporangium is called microsporogenesis. The process of conversion or formation and development of microspores from functional microspores from microsporangium is called a microsporogenesis. Formation of microsporogenesis. And like that, the megasporangium producing megaspores is known as two processes, microsporogenesis and megasporogenesis. Microsporogenesis and megasporogenesis. Two processes, just like the spermatogenesis and oogenesis in animals. See. By heart then and there. See, next is stamen, anther, pollen grains. Microsporophyll, stamen, anther, pollen grain. Stamen is the microsporophyll. Anther is the microsporangia. Pollen grains are the microspores. Stamen. This is a stamen. Stamen is also known as microsporophyll. Anther, connective, stalk or filament. This is stamen. See, this is the proximal end and this is the distal end of the filaments. See, stamen is also known as microsporophyll. It contains sac-like structure. Called anther. Stalk of anther that connects into the flower. Or thalamus of the flower is called filament. Young anther is a rectangular mass of tissue. This is the young anther. So, sticky ice structure. While we take the cross section, it will be seen as a rectangular mass. The formation of four constriction, 1, 2, 3, 4 constriction, one opposite to the other, make them bilobed. Each lobe is dithecous, connected by a connective. Make them bilobed, dithecous and four-sided structure. So here 4 side, 1 side, 2 side, 3 side, 4 sides. 4-sided. The mature anther is bilobed, each lobe is dithecous because at the four corners there developed mass of tissue. This is mass of tissue which forms microsporangium or pollen sac later, in which the pollen grains are produced. So sporogenous tissue are developed. Microsporogenous tissue are developed at the four corners. Systematically, there is the formation. It is not given in a textbook. The mature anther is bilobed. Each lobe is dithecous. 4 theca present, hence it is called dithecous. Dithecous by 2 lobes. Dithecous or bidithecous or didithecous. Anyway, bidithecous, bilobed, 4 pollen sacs or microsporangia, 4-sided with 4 constrictions, one opposite to the other. That is about the general account. So you know, at the 4 corners there develops 4 groups of sporogenous tissue. They are actually microsporogenous tissue. Means from which you know the microspores are produced after meiosis. Okay? Anther is covered and protected by anther is covered and protected by 4 volutes. Anther is covered and protected by 4 volutes. 4 volutes are there. So they are epidermis, endothelium, middle layer, and tapetum. Epidermis is outermost single layered with elongated parenchymal cells as in the epidermis of other organs of the plants. Endothelium, second layer, single layered, middle layer, 3 to 5 layers. 3 to 5 layers constitute middle layer. Innermost layer, single layered, cenocytic and nutritive. See, there are 4 layers to form the anther wall. The epidermis, the outermost layer, it is single layered. Endothelium, the second layer, also single layered. Middle layer, 3 to 5 layers constitute a middle layer. Tapetum, the innermost layer, single layered, Sinusoidic, multinucleated, binucleate to multinucleated, and considered as nutritive layer, which enclosing the sporogenous tissue, microsporogenous tissue. See, this is the cross section of a cut end, anther wall, sporogenous tissue, so connective, and this is anther wall. This anther wall is connected, anther wall is enclosing the sporogenous tissue at the four corners of the tissue of the anther. Okay, then in details. This is the outermost boundary, the epidermis, formed of elongated parenchyma cells without any intercellular space. They are thickly packed, no chlorophyll, as the epidermis of other worlds or other parts. This is epidermis. Epidermis, and covered and protected by a layer of cutin called a cuticle. So, a layer with the cuticle of another wall, that is the epidermis. This epidermis. Inner to that, there is a single layer of more or less large cells prominent nucleus is there, but with a prominent nucleus. This is endothelium. Endothelium. Then middle layer. So here you know tapetum. This is tapetum. This is multinucleated protective covering of tapetum. And here 1, 2, 3 to 5 layer, more or less 3 layer. This is inside, you can see sporogenous tissue with anther wall. This is cuticle sporogenous tissue. Epidermis, endothelium, middle layer. Tapetum. Okay, so this is the enlarged view of a part of anther lobe. Formerly I told you epidermis, endothelium, middle layer, tapetum, which are the layers of anther wall, constitute the anther wall. So anther wall contains 4 tiers of cells. 4 tiers of cells. So cuticle, outermost layer epidermis, then inner to that endothelium, then middle layer, tapetum, then sporogenous tissue. Which later becomes pollen. These are actually modified as pollen mother cells. So here you know the connective, this part. So cuticle, the protective covering of cutin layer. The cuticle is the protective layer of epidermis. As a whole we can say the anther wall. It is formed of a single layer of cells which are elongated and thickly packed without any intercellular spaces. Inner to this, see, this is only a single layer. Inner to this, more or less enlarged cells which are provided with a prominent nucleus. That is called endothelium, the second layer. That is also single layered. So the inner to the endothelium, there is the middle layer formed of 3 to 5 layers. Here already indicated only 3 layers. 3 to 5 layers. Then tapetum, the innermost layer, the nutrity layer. So some key points in connection with these parts. So you know, the tapetum encloses sporogenous tissue, or they are modified as pollen mother cells, written as PMC, or microspore mother cell, which are all diploid. 2n number is there, which are all diploid. See, cutinized wall layer. Some key points. Cutinized wall layer of anther. Epidermis. Then the layers which help in the rupturing of anther wall. Epidermis, endothelium, middle layer. Innermost layer of anther wall. Tapetum. Cenocytic layer of anther wall, also tapetum. Nutrity layer of anther wall. Tapetum. Protective covering of sporogenous tissue. Protective covering of sporogenous tissue. Protective covering of sporogenous tissue is also tapetum. Which will give nourishment, and moreover some enzymes like some rupturing enzymes, cellulase like enzymes will be also produced inside this nutrity layer. See, some key points in connection with the anther wall. Cutinized wall layer of anther, epidermis. The layers which help in the rupturing of anther wall when they attain maturity. Epidermis, endothecium, then middle layer. The innermost layer of anther wall, tapetum. Cenocytic or multinuclear layer of anther wall, tapetum. Nutritive layer of anther wall, tapetum. The protective covering of sporogenous tissue is also tapetum. And moreover, the layer which disintegrates when the anther attained maturity, that is also tapetum. So in mature anther wall or in mature anther, there is no distinct layer. The tapetum is not seen because they are disintegrated for the nourishment of the production of pollen grains or the microsporogenesis. During the microsporogenesis, the nutritive part of tapetum which are stored in this layer is utilized for the growth and development of pollen grains. Okay, and so that is about anther wall. Okay, then next is the microsporogenesis. 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 The formation and development of microspore or pollen grains from diploid pollen mother cells or microspore mother cells after meiosis is called microsporogenesis. So microspore mother cell or pollen mother cell, PMC. Megaspore mother cell is also MMC. Microspore mother cell is also MMC. To detect the microspore mother cell, we can say PMC we have to write. The formation and development of microspore or pollen grains from diploid pollen mother cells or microspore mother cells after meiosis is called microsporogenesis. So what is microsporogenesis? Once again, the formation and development of microspores or pollen grains from pollen mother cells or microspore mother cells after meiosis is called microsporogenesis. So the resultant product of microsporogenesis is the pollen grains. That means that is pollen grains. Then pollen grains heading. The formation and development of microspore or pollen grains from diploid pollen mother cells after meiosis is called microsporogenesis. The resultant product of microsporogenesis is the pollen grain. Then structure of pollen grain, also known as microspore. Study of pollen grain is called palynology. Pollen grains are very small, more or less globular, sticky or spinous, having 2 to 5 micrometer in diameter. What do you know about pollen grains? Study of pollen grains is called palynology. Palynology, study of pollen grains. Pomology, study of fruits. Nidology, study of nest. Paleontology, study of fossils. Anyway, study of seeds, spermology. Study of flower, anthology. So many branches will be there. Among them, palynology is the study of pollen grains. Study of pollen grains is called palynology. The pollen grains are very small, more or less globular in appearance. Sticky or spinous, having 2 to 5 micrometer in diameter. See, this is a pollen grain. It is very small. Though it can be seen with our naked eye, it is very so more or less small. We can see with our naked eye, according to hibiscus rosa sinensis, when the flower is come in touch or in touch with our skin, the pollen grains are deposited, as it is sticky as well as spinous. See, very small, we can see, not so microscopic, but we can see with our naked eye. So, structure of pollen grain, and so structure of internal structure of pollen grain. See, listen. Each pollen grain is bivolate, bicelled, binucleated, with one or more germpore. See, each pollen grain is bivolate, that is indine and exine. Bicelled, binucleated, that is the mature pollen grain. Bivolate, exine, bicelled, weighted cell and generative cell, binucleated, two nucleus and generative nucleus with one or more germpore. So, microsporogenesis followed by structure of mature pollen grain. This is PMC, diploid. It undergoes meiosis, reduction division or meiosis, forming 1, 2, 3, 4 nucleated, which is haploid. And then it is wall appears. Wallets, wall of pollen mother cell. Arranged in tetrahedral manner. Wall layer. tetrahedral tetrad. 3 wicks dice sweets. 3 placed or 1 placed over 3 underneath. See, 3 at the base and 1 is placed on the top. So that it becomes tetrahedral nucleus. Then they get separated each other and becomes more or less globular. By rupturing bi-celled mature pollen grain is coming out. This is microsporogenesis, formation and development. This is haploids. Here reduction division takes place, haploids. Thus pollen grains are formed from one pollen mother cell. 4 pollen grains are formed. See, that is the microsporogenesis. See, mature pollen grain is bivolate, having exine and egg as the volates, bicelled vegetative cell and generative cell, and concerned nucleus concerned tube nucleus or vegetative nucleus, generative nucleus in the generative cell with one or more germ-pore. Mature pollen grain. The pollen grain is more or less globular, and that is why there is no longitudinal section or cross section. A section. So this is a section. Mature pollen grain is, so study about pollen grain is palynology. Then mature pollen grain is usually bivolate. So this is outer wall layer, exine. This is inner wall layer. Exine. Inner wall layer is intine. Exine and inner. This is outer wall layer. This is inner wall layer. exine is very hard, maybe sticky or spinous, and that is due to the presence of a chemical substance called sporopollenin, red colored sporopollenin present, formed of sporopollenin. And hardness as well as the sticky nature will be due to the presence of this chemical substance called sporopollenin. It gives protection, resistance to the microbial activities. So sporopollenin gives protection, resistance to the stress conditions, having antimicrobial activity, activity. See, protection, resistance to the stress conditions, having antimicrobial activity. These are the characteristic feature of sporopollenin, and in addition to that, it gives hardness to the wall layer, outer wall layer, and it is thick. Wall layer is thick. When compared with the inner wall layer. That is, we can say the chemical substance that present in the exine is sporopollenin. Next is inner layer. It is thin, formed of cellulose and pectin. Thus, two wall-layered exine and indine. It encloses, or indine encloses, a large cell which is called tube cell, or vegetative cell. Enclosing, there is a common cytoplasm. Common cytoplasm is there, and this is the nucleus, which is irregularly round, not smooth round. That is called tube nucleus. Tube cell enclosing tube nucleus, irregularly round. Round in appearance, not smooth. Then the other cell, this is common cytoplasm. Common cytoplasm is there, and within this cytoplasm there is a floating cell. Within this there is a floating cell called generative cell. carrying generative nucleus. This is cell. Cell is also spindle shaped, and the nucleus is not so round, and it is also spindle shaped in appearance. So thus it is two celled. There is a thin membrane cytoplasm, that is common cytoplasm, covered and protected by this plasma membrane encloses the common cytoplasm, within which irregularly round large nucleus, and the vegetative cell is large, generative cell is very small. Thus it becomes bicelled and binucleated. Then within the wall layer there is a thin region. This is where sporopollenin is absent. So this is the germ bore. Germ bore. Pollen grain is more or less globular in appearance, having two wall layers, namely exine and indine. Exine is the outer wall layer, indine is the inner wall layer. Outer wall layer contains a chemical substance called sporopollenin. Outer wall layer is very thick. Thick wall. It gives protection, resistance to the stress conditions, having antimicrobial activity. Sporopollenin. Indine, the inner wall layer, formed of, which is very thin, formed of cellulose and pectin. It encloses the plasma membrane, which carries a common cytoplasm, and the large cell is called the vegetative cell or tube cell. It contains tube nucleus, which is irregularly round. Within the common cytoplasm of the vegetative cell, there is a spindle-shaped cell called a generative cell, carrying generative nucleus. So, two nuclei, binucleated, two cells, vegetative cell, then generative cell, that is bicelled, exine, indine, biwall layered, and one or more germ bore. Here in dicots, one germ bore is there, but in monocots it may be 1 to 3. And the significance of germ bore. So listen please, significance of germ bore. So there are 4 significant features in connection with the germ bore. First is the thin area of wall layer of pollen grain, germ pore. The part which is not differentiated into exine or indane in the pollen grain, that is germ pore. The region where sporopollenin is absent, germ pore. The region through which the pollen tube is produced. So after pollination, the pollen germination takes place to the stigma. So this figure showing the germ pore producing pollen tube to the stigmatic region of the carpel. And so germ pore is the thin area of the wall layer which is not differentiated into exine or indine. Sporopollen is absent there. It is a thin region through which pollen tube is produced, and the part which is not differentiated into exine or indine. Thin area of the wall layer is pollen. Wall layer of pollen grain is germ pore. And these are all about the significance of germ pore. Then, at the time of liberation of pollen grains from anther, about 60% of pollen grains are liberated from the anther is two-celled. Tube nucleus and generating nucleus. Whereas 40% of pollen grains are liberated from the anther is three-celled. Two sperm cells plus a tube cell. Three-celled. Whereas this is two-celled. Tube nucleus, tube cell and generative cell. And that is about the pollen grains. Remember please, sporopollenin, thick walled exine is provided with a chemical substance called sporopollenin. It gives hardness, prevention of stress conditions, having antimicrobial activity, thus protection of the inner part of the pollen grain, which are by the presence of sporopollenin. Like that, the nutritive tissue, that is the anther wall layer, nutritive in function, innermost layer, cenocytic, covering of spore, microspore, mother cells or sporogenous tissue in the anther, and when the anther wall or when the anther attained maturity, it will be disintegrated, the tapetum, the most tissue. Here also 4 comments. And these are all about general account of pollen grains. In addition to that, the pollen viability, pollen products. In connection with pollen grains, you have to know the pollen viability, pollen products, pollen allergy, pollen bank, etc. Pollen viability. So you know viability means the pollen grains remain functional. So the period of pollen grains that remains functional is called pollen viability. Then some pollen products will be there. As the pollen grains are nutritive, some products will be there. That they are all seen on the next class. Pollen allergy, parthenium, it is very important. And pollen bank, just like the storage of pollen grains, because a crop improvement program for crossbreeding, there is a storage of pollen grains in a liquid, frozen state in a liquid like nitric acid, 195 degrees centigrade, minus 195 degrees centigrade, 96, 95 for insemination, 7 collections. And that these are all can be seen on the next class. Pollen viability, pollen product, pollen allergy, pollen bank. These are all about on the next class. Structure, the figure is given in the theory classes. You have to label they are asked. And moreover, you have to know the functions, etc. Okay, rest on the next class.